Bismillahir Rahmanir Rahim. LL37, the only member of the human cathelicidin family, has antiviral, antifungal, and antibacterial properties. Its complexes with nucleic acids have been found to stimulate the immune system, positively affecting the human innate immune response. However, on the other side, research indicates that under certain conditions, LL37 nucleic acid complexes may contribute to the development of autoimmune disorders such as psoriasis and systemic lupus erythematosus. Welcome again to Derma Immunology. I'm Naglaa Elmongy, Professor of Dermatology, Cairo University, and our video today is LL37's complex dance with nucleic acids. The innate immune system serves as a vital first line of defense against invading pathogens. Various classes of antimicrobial peptides play specialized roles in protecting the body from a wide range of microbial infections. LL37, the only cathelicidin peptide in the human proteome, is one of the most significant and extensively studied members of this group. It shows broad-spectrum antiviral, antifungal, and antibacterial properties along with antibiofilm activity. LL37 has been found to form complexes with various nucleic acids, including mitochondrial DNA, genomic DNA released from neutrophils through neutrophil extracellular traps or nets, and RNA. These LL37 nucleic acid complexes have been shown to activate toll-like receptors such as toll-like receptors 3, 7, 8, and 9, which are critical components of the innate immune response. And while complexes of LL37 with nucleic acids are known for their beneficial immunomodulatory effects, several studies have also highlighted their role as a disease contributors with harmful impacts on human health. As regard LL37 structure, the peptide is composed of 37 amino acids and adopts an alpha helical structure. An alpha helix is a coil configuration formed by a sequence of amino acids in a protein. It has spanning residues from 2 to 31 with a disordered tail at the C-terminal in a physiological solvent environment. The active LL37 peptide is produced through extracellular cleavage of the peptide from the precursor protein human cathelicidin, a process mediated by proteinase 3 in neutrophils or serine proteases in keratinocytes. LL37 is employed by neutrophils, macrophages, epithelial cells, keratinocytes, B cells, and natural killer cells to target and eliminate infected cells. This peptide is immunostimulatory and plays a key role in inflammation and the destruction of pathogens as well as infected host cells. The expression of LL37 is regulated by factors such as vitamin D3 and butyrate and can be induced by infection, physical injury, or an endoplasmic reticulum stress response. Butyrate is a short-chain fatty acids produced by good bacteria in the large intestine through fermentation of dietary fibers in the lower intestinal tract. LL37 is essential for human health, and its upregulation has been proposed to play a significant role in decreasing the severity of various infectious diseases, including severe cases of coronavirus disease, COVID-19. LL37 also promotes cellular regeneration by directly participating in autophagy and boosting the chemotaxis of progenitor cells during wound healing. Normally, unbound peptide would quickly be degraded by proteases. However, due to its ability to form complexes with various biomolecules in solution, such as plasma proteins, amyloid beta, and lipid bilayer phospholipid membranes, LL37 is effectively shielded from proteolytic degradation. Additionally, LL37 can form complexes with F-actin and glycosaminoglycans. LL37 has a positive net charge of +6, which facilitates electrostatic interactions with negatively charged molecules and structures such as phospholipid membranes and various types of nucleic acids. And as I already said in the introduction, LL37 has been shown to form complexes with various nucleic acids, including mitochondrial DNA, genomic DNA released from neutrophils in neutrophil extracellular traps, and RNA. These LL37 nucleic acid complexes have been found to activate toll-like receptors such as toll-like receptor 3, 7, 8, and 9, all of which play a key role in the innate immune system. And again, while complexes of LL37 with nucleic acids are known for their beneficial immunomodulatory effects, several studies have also identified these complexes as contributors to diseases having harmful effects on human health. Studies have demonstrated that LL37 penetrates cell membranes and is internalized by human macrophages and osteoblasts through endocytosis. Endocytosis is considered the primary pathway for membrane translocation, while general cytoplasmic delivery and endosomal escape do not appear to be efficient mechanisms. Now I'm going to talk about LL37 nucleic acid complexes. Several studies have shown that LL37 has an affinity for binding to DNA forming complexes that are important for the human innate immune system. The helical double-stranded structure of genomic DNA has a negatively charged backbone due to its phosphate groups, making it an ideal binding partner for the cationic LL37 through electrostatic interactions. To fully understand the mechanism of action of LL37-induced nucleic acid condensation, in-depth biophysical structural studies are required. Extensive research in this area has shown that LL37, when complexed with DNA, leads to a greater induction of interferon alpha production in dendritic cells, an effect associated and related to the structure of toll-like receptor 9. As I have said before, LL37 is used by natural killer cells to target and eliminate infected cells. A study found that complexes of LL37 with DNA subunits can also promote the proliferation and activation of natural killer cells in the peritoneal cavity, suggesting a unique role for these complexes in the innate immune system. Evidence that LL37 DNA complexes increase the number of natural killer cells within a biological system is important, as natural killer cells not only eliminate infectious agents and have antimicrobial properties, but they are also essential in the fight against cancer. In researches also, LL37 demonstrated its potential to protect nucleic acids from degradation by nucleases. Beyond the beneficial effects that these complexes seem to have on the human body, LL37 and its ability to bind DNA play a key role in activating plasmacytoid dendritic cells in psoriasis. LL37 and DNA form aggregated and condensed structures, transforming inert self-DNA into a trigger for interferon production. This suggests that LL37 can disrupt the innate tolerance of self-DNA, a process that was believed by some authors to contribute to the development of autoimmunity in psoriasis. Now let's talk about LL37 mitochondrial DNA and inflammation. Inflammation is commonly associated with oxidative stress and damaged cells, which release elevated levels of molecules known as damage-associated molecular patterns into their surrounding environment. Mitochondrial DNA has been identified as damage-associated molecular patterns and is recognized as a key factor in activating the innate immune system. It's also primarily known for its role in causing maternally inherited mitochondrial diseases. Mitochondrial DNA is located in the mitochondria and encodes vital protein subunits required for oxidative phosphorylation. It is a small double-stranded circular molecule consisting of 16,569 base pairs and encode for 37 genes. Mitochondrial DNA regulation is thought to be primarily governed by cell-specific mechanisms and in response to intrinsic or environmental stresses. However, the high abundance of mitochondrial DNA in many cells and tissues suggests that other factors may contribute to its elevated presence. Recently, mitochondrial DNA has gained increasing attention as a potential activator of human receptors affecting antimicrobial responses and inflammatory conditions within the innate immune system. On the other hand, abnormal responses of the innate immune system are closely linked to conditions such as autoimmune diseases, metabolic syndrome, and neurodegeneration. Given that mitochondrial dysfunction and/or damage is a common feature in these diseases, the upregulation of mitochondrial DNA may either contribute to or exacerbate these pathologies. The literature suggests that naturally upregulating LL37 could help to maintain low mitochondrial DNA levels. For example, mitochondrial DNA has been identified as an inflammatory mediator in cardiovascular diseases and atherosclerosis. Given the LL37's ability to form complexes with nucleic acids, it's logical to assume that increasing LL37 levels would provide more binding partners to eliminate mitochondrial DNA, preventing it from stimulating receptors and triggering immune responses. This complexation of LL37 with mitochondrial DNA could in turn help to reduce inflammation associated with cardiovascular diseases and atherosclerosis. However, in a study done by Zhang et al., they demonstrated that complexes formed between LL37 and mitochondrial DNA may actually promote atherosclerosis. They discovered high levels of LL37 bound to mitochondrial DNA in atherosclerotic plaques and plasma, which contributed to increased atherosclerotic lesions in apolipoprotein E deficient mice. Also, these complexes were resistant to autophagy and appeared to protect mitochondrial DNA from DNase to degradation. Despite this, toll-like receptor 9 still recognized the mitochondrial DNA, triggering inflammatory responses that activated autoimmune processes, including the release of chemokines and cytokines. This suggests that complexes of cathelicidin and the mitochondrial DNA within a cell are difficult to clear, potentially leading to sustained activation of toll-like receptor 9 signaling pathways. Treatment with antibodies targeting the LL37 mitochondrial DNA complex reduced lesion formation, leading the authors to conclude that these complexes contribute to atherosclerotic plaque formation. Disrupting these complexes could therefore provide potential strategies for preventing and treating atherosclerosis. Another study demonstrated that by inhibiting mitochondrial DNA degradation induced autophagy, LL37 mitochondrial DNA complexes exacerbate local inflammation in sepsis-induced acute lung injury. Similar to the atherosclerosis study, higher levels of LL37 were found in the serum of severe sepsis patients compared to those with mild sepsis. As a conclusion from the first video about LL37 complex dance with nucleic acids, complexation of the human cathelicidin LL37 with various types of nucleic acids has been shown to form diverse structures, exert immunostimulatory effects, while also causing harmful effects. Lastly, I hope that you have enjoyed this video And I'm reminding you again to subscribe to the channel and activating the alarming bell. And thank you.